Welcome to the Code of Life. I'm Randy Neal. CRISPR was recently in the news because the scientists who discovered it just won the Nobel Prize for Chemistry. The Nobel Prize Committee describes CRISPR as genetic scissors, an editing tool for rewriting the code of life. Using CRISPR, researchers are contributing to new cancer therapies, curing inherited diseases, developing mold resistant plants and crops, or, like our two guests today, working to restore sight to children and helping people with deadly brain tumors live longer. CRISPR is a game changer when it comes to scientific breakthroughs, and our guests today will also change the world with their research and study into incurable diseases. They both have resumes and accomplishments too long to list, but Dr. Elizabeth, or Beth Simpson, is a world-leading scientist in mammalian genetics and genomics. Her overall research is to improve treatment for human disorders of the central nervous system. She's currently using CRISPR to help cure a congenital blindness called aniridia. And Dr. Sheila Singh is a world-leading scientist in human cancer stem cell biology, focusing on alleviating brain tumors, glioblastomas, like the one that killed the tragically hip Squirt Downey, using CRISPR to investigate genetic abnormalities of brain tumor initiating cells with the ultimate goal of better prognoses for people with brain cancer. Welcome to both of you. Thanks so much for joining us.
Thank you for having us.
Let's start with, what is CRISPR exactly and why is it so revolutionary? Beth, let's start with you.
So CRISPR is an acronym for clustered regularly interspersed short palindromic repeats. So it's a mouthful, let's just call it CRISPR. And it comes to us from bacteria. So bacteria have an adaptive immune system where a bacteria can be invaded by a virus just like humans can be invaded by a virus. And they need to protect themselves. And one of their mechanisms to protect themselves is CRISPR. And it's a mechanism to cut up the DNA of the invading virus. And the Nobel Prize winners, they collaborated together. Two women scientists working separately came together and published a paper in 2012 that not only further described the bacterial system of CRISPR, but really recognized for the first time what an incredible tool it could be if we moved it into mammalian systems and used it to manipulate the genome of plants and animals and even humans.
Sheila, how did these two women get to this point? I mean, they were both on different sides of the world and they really collaborated, didn't they, to come up with this research?
Well, I think the wonderful thing about the Nobel Prize winners is that they built on each other's discoveries. And I think that they were very keen to learn from each other. And they both had important roles in the discoveries that they made, that this bacterial enzyme could actually have other uses and essentially be repurposed, as Beth had mentioned. So collaboration in science is really important. It enabled the Nobel Prize winners' work enabled other scientists like Feng Zhang and George Church to go on and make similarly groundbreaking discoveries in mammalian cells. And then that in turn enabled researchers like Beth and I to appropriate these amazing tools and use them to explore and make new discoveries in our own areas of research. So again, a long chain of discovery.
Let's talk a little bit more about your research. You mentioned when we spoke earlier that collaboration is a big part of your research, but exactly what are you using CRISPR for in your study of glioblastomas? And tell us a little bit about your history.
Of course. So I am running a large brain cancer laboratory at McMaster University. I'm also a pediatric neurosurgeon at McMaster Children's Hospital. So I'm extremely interested to uncover the molecular mechanisms of very difficult and hard to treat brain cancers that are currently failing conventional therapy like chemotherapy and radiation therapy. So I have the fortune of being able to see what happens when patients are afflicted with brain tumors, and to really have a good understanding of what they go through, and then to come back to the lab and to try to develop model systems that accurately replicate what happens in human beings. And so we are fortunate to be able to take cells from patients' brain tumors and study them in the lab and engraft them into mice who don't have an immune system in order to study and model the growth of brain tumors. And so we were lucky enough to work with collaborators who have a very different expertise from me, like Dr. Jason Moffat at the University of Toronto, whose expertise is in using functional genomics tools like CRISPR. So we were able to get his CRISPR libraries and drop them on our brain cancer cells. And this allows individual labeling of every cell, every gene that's expressed in the brain tumor. And then as we study how the brain tumor grows and evolves over time, and how chemotherapy and radiation delivered to the mice, our little mouse patients, how that changes the expression of these genes and their ability to drive brain tumor formation, at the end of this grand experiment, the CRISPR screen, as we call it, enables us to tell exactly which genes are essential drivers of the growth of that patient's brain tumor. And so you can imagine we could even develop this work in a personalized fashion, because maybe every patient's brain tumor has a different essential driver. But CRISPR as a screen enables us to determine those key genes that are driving the growth of brain tumors, and then we can develop new therapeutics to block those targets.
And Beth, you're using something similar, and in this case, potentially, which I find amazing, you're looking for a cure for a certain type of congenital blindness.
I it's CRISPR again in all its different new forms. I was at a genomics meeting. I was speaking at a genomics meeting in Italy, in Rome, and Dr. Charpentier, the Nobel Prize winner now, stood up and gave her talk. And I can remember it was just jaw dropping. I remember whispering to my neighbor next to me what I could do with that. And it really impacted all the research. That was 2013, impacted all the research going on in my lab. By far the most exciting is a recent paper by a graduate student. Trainees are incredibly important in research labs. And this is a paper by Zainab Mohanan. And she showed that we could, for aniridia means without an iris. And that's what the patients or the parents notice first. The iris is the colored part of your eye. And in these children who have an inherited blindness, the iris is missing or very small. And so the pupil looks really large and the eyes look really dark. But unfortunately, the gene that is mutated, PAX6, is a master regulator of eye development. So it's not just the iris, it's the entire eye, which is developing abnormally. And the children are born with poor vision, and over time will lose their vision completely. And so we're interested in using CRISPR to change the base pair that is mutated back to normal and turn that gene PAX6 back on in these children's eyes. To start with, we had to make a better mouse model, because in the area of gene therapy, the mantra is to cure the mouse first. So we're working on curing the mice, and the work is ongoing, but we've just built our therapy into viruses, and it's actually with viruses we will deliver it to the eyes of these mice and hopefully improve their vision, laying the foundation for a clinical trial in children.
And how far away do you think we are from that?
You know, you asked me about that, and I decided to go look up the leading CRISPR therapy in my field, which has the really cute name, Edit 101. And Edit 101 made their mouse model, published their mouse model in 2013. They then published curing their mouse model in 2019, and they have treated their first patient in 2020. So these things can move very quickly.
I wanted to ask you that as well. how do you think CRISPR will potentially change medicine in the next 5 years, 10 years, 20 years? But I also want you both to talk about the ethics of this. We're talking about studying mice, we're studying about CRISPR, which is a gene editing tool, and some people have problems with genetically modified organisms. I want you both to kind of comment on that. And Sheila, we'll start with you.
So clearly, all of the research we do, especially speaking for Canada, we have really strict and well laid out ethics guidelines for almost everything we do in terms of animal research, and certainly in terms of human research, meaning running clinical trials where we're offering experimental therapies to people. And ultimately, the CRISPR technology will have to be governed by similar agreed upon rules of ethics. And there are global bodies that are starting to establish these rules. But the one thing that will be interesting to watch is, as Beth mentioned, in the old days before CRISPR, it took much longer to make these advances. And now that we're seeing this accelerated rate of success, we're going to have to run even faster to keep on par with the ethics rules. I guess I wanted to distinguish between two types of gene editing. You can gene edit what we call the germline, which is cells that are actually in embryos that make every cell in the human body. And that's of much greater import and consequence than tissue specific cell editing, which means that you're taking cells from a certain lineage and you're genetically engineering changes in those cells. So there are different implications between those two types of interventions that will have to be governed by different sets of rules and ethics. And perhaps the challenging thing will be to find international consensus in that regard, because every country has a different board of ethics and set of rules.
Beth, do you want to jump in?
I agree with everything that Sheila just said, and I just wanted to mention that I think the world has agreed that germline gene editing in humans is not appropriate, and in many countries illegal. And the only person that we know of who has done that is in jail, and so that's totally appropriate. In animal research, we have been able to edit the germline of animals and plants for a very long time. And so, although CRISPR is game changing in animals and plants in the speed and accuracy at which we can modify the germline, it isn't actually different. And so, the ethics considerations are well established and well underway and are further advanced for animal and plant research, and a little bit more established than what we've seen there in human. What I would like to mention is that there's also two types of CRISPR. There's CRISPR where you remove the cells from the body, and there's just two amazing studies going on, one in Europe and one in North America, for sickle cell anemia and beta thalassemia, both very terrible blood disorders, where they remove the cells, they CRISPR them in the laboratory, and they're able to characterize them greatly prior to putting them back into the patient. And those kinds of approaches received permission to do clinical trials first. The one that I mentioned, which is an in vivo introduction of CRISPR into the eye of young people who are going to go blind, only received permission to go ahead last year. And as I mentioned, the first patient this year. So I do think we are appropriately moving slowly and carefully, but with knowledge that these are incurable diseases desperately in need of treatments. And so clinical trials are being allowed in some cases.
Just a couple of minutes left, and I do want to leave it with this final question. Where do you think that CRISPR will take us in the next 20 years?
If I jump in just for a second, more Nobel Prizes. Oh my goodness, this is only the first one. I think the people who now use CRISPR to develop therapies will be groundbreaking, and we will see more prizes in the future in the CRISPR domain. Sheila, what do you think?
I agree completely, Beth. I can think of so many applications that are going to emerge over the coming years, and the one that I'm particularly interested in is the combination of CRISPR gene editing and immunotherapy. So, immunotherapy is a relatively new form of cancer therapy that harnesses the body's immune system to try to specifically target and kill cancer cells. And you can actually take patients' T cells, which are the soldiers of the immune system, and you can CRISPR them to actually enhance their ability to attack specific targets on the surface of a tumor cell. And we've just had our first clinical trials showing that you can CRISPR edit T cells, give them to a patient, and this is both safe and non-toxic. So I can only imagine what's going to happen with immunotherapy and our ability to specifically target tumor cells in cancer patients over the next 5 to 10 years.
Wow. That just gave me goosebumps. Okay, we're going to leave it there. Scientists for the win again. Dr. Simpson, Dr. Singh, thank you so much for your time and for doing what you do and tackling these incurable diseases to make us all healthier.
Thank you for speaking to us today.